Assalamualaikum and good afternoon to Dr. Sharum, Dr. Rosina and Prof. Arif. My name is Nova Zelina binti Ahmad. I'm the representative from Group 3, and for today's presentation, we are from Group 3. We'll be presenting on genetic resistance to disease and parasite infestation in livestock. Moving on to the introduction, as we all know, parasite and disease affecting livestock can have a significant effect on the animal production, productivity, human health, and overall process of economic development. And the application of established method for disease control most frequently have negative consequences and have raised some concern, which is such as the evolution of resistance to the treatment applied, food safety related issues and such. So another alternative application is to promote the use of livestock breed that are genetically tolerant or resistant to these diseases. So by definition, resistant is the host's ability to resist infection or moderate parasite life cycle, and for tolerant, the host's ability to withstand infection and disease effects. As for the genetic resistance, is a genetic component to resist pathogen infection. That's all from me, and I would like to pass to continue the presentation.
Okay, thank you very much, Fazina. My name is Siti Suhaila, and I will explain about the concern or challenges regarding disease resistance. First of all, it is very difficult to identify the phenotype that is responsible for disease resistance. This is because not all healthy animals are susceptible to the disease, and the success of the selective breeding is highly dependent on the in identifying the phenotype. Secondly, in selective breeding for disease resistance in livestock, it may not be ethical and cost efficient to challenge each livestock animal with a pathogen to determine its level of disease resistance. And lastly, it may disturb the homeostasis of the immune system of the animals itself. The development of highly resistant animal to one specific pathogen may make the animals more susceptible to another pathogen. Okay, then I will explain about the importance of genetic resistance. First of all, it acts as an alternative to improve livestock products, and then it saves treatment costs. We can avoid the economic cost of the disease. Then it also genetically modified animals provide valuable models. These animal models can be used to investigate disease progression. Lastly, with genetic resistance, we can control the disease. Controlled breeding of the plants and animals without genetic diseases will sooner or later eliminate genetic disease from the future offspring. That's all from me. I will pass to Priya.
Okay, now I'll be talking about disease and studies that have been conducted on genetic resistance to the diseases. Okay, in cattle, the common diseases that have been found is foot and mouth disease, mastitis, tuberculosis, and brucellosis. So now I'll be taking this example, which is mastitis, which is the inflammation of udder that affects this milk yield, and it's a very major problem in the dairy farm. So what the researchers done, that they took this candidate gene that is really important in this complex process of this genetic resistance to the mastitis. So we have BOLA, which is bovine leukocyte antigen complex, and this variability in this BOLA, it gives this opportunity in identifying these alleles that is really significant in expressing the resistance to this mastitis. Apart from using this BOLA, we can use somatic cell count as well. So if you see this more than 300,000 somatic cell count, it indicates that the cow's milk is affected by mastitis. So moving on to the sheep and goat. So the common diseases in the sheep and goat is the scrapie, foot rot, and mastitis and dermatophimosis. So example that is used in this case is foot rot. So this foot rot is the inflammation at the hoof part where it causes really painful for the sheep or the goat. So this also affect the productivity of the goat, as it will affect the milk quality and the meat quality as well. So the 26 genes that is found in two gene loci of this OLA complex is responsible for this resistance to this food rot problem. So if the researchers increase the number of these 26 alleles at the OLA complex, it can help to prevent this food rot disease. So moving on to Rick.
I'm going to explain more about the genetic resistance to parasitic infections. Because of the domestications of the animals, so the animals tend to break the bonds between the animals and parasite bond, in which the animals now are more susceptible to the parasitic infections. And then absence of the compensatory food, and there's no selection effort of the farmer, in which this will cause the parasitic infection to be less resistant to this animal, because there is no selection, as the animal that are more susceptible, they are put into breeding program. And then the variability of resistance in flock, in which there will be animals in which with a high resistance and low resistance. The low resistant animal, they will tend to be more susceptible to the parasitic infection, and then they will tend to have a more parasitic infection, and then the more level 3 larvae produced, and then this will cause contamination of the pasture and complete the cycle. And when there is parasitic burden in the farm, the production decreases, and there will be poorer carcass, and you need more antibiotic treatment, and this will incur more cost. And then more anti-helminth treatment will have more antibiotic resistance to these worms, and then they will contaminate the pasture, and then completing the cycle with susceptible animal. And the demand of the drugs-free meat from the consumer cannot be achieved, because we tend to use a lot of anti-helminth treatment. Next. So this is a research about the parasitic infections, in which this will be focused upon mother buffaloes, deers, cattle, goats, and sheep. So for the deer, in which we can find in Malaysia, is the samba deer, which is more resistant to tick infections. So next.
Okay, hi, my name is Shabika Betty Nasruddin. So I will explain more about the solutions and the new technology for this disease resistant and parasite infestation in livestock. So the first one is transgenesis. Transgenesis is transfer of transgene between organisms, so the transgenes can be carried by their offspring. So the transgenesis will pose various resistance to disease. And the next one is molecular breeding. Molecular breeding is application of molecular tools in animal breeding, and in this case we use marker assisted selection, which is there's 4 types of marker, which is morphological, biochemical, molecular, and many more. So next I will pass to.
Okay, thank you, Shahnaz. My name is Siti Anissa binti Nordin. Now I'll explain the future strategy in breeding for disease resistance. First, the farmer should be aware in the use of therapeutic agents, as it might lead to the occurrence of antibiotic-resistant bacteria or pathogen in the farm. The farmers can change the type of antibiotic that they use frequently, or reduce the use of antibiotic in the farm to prevent the bacteria from being resistant. And next, the antibiotic-resistant bacteria have the possibility to be transferred to human by ingesting the infected meat. Okay, next, the farmers can practice biological control. Farmers can use spectrophages as in the control of pathogen, for instance, E. coli and meningitis in chickens and cow. These spectrophages will target a specific bacteria and attract it and attack it, instead of using broad-spectrum bacteria that may kill normal flora in the animal's body. And lastly, the farmer can use selection pressure for general immunological competence and targeted resistance tests for specific major pathogens, for instance, using marker-assisted selection on candidate gene that has been explained by Shahnaz previously. That's all from me. I'll pass to Faz for conclusion.
So as a conclusion, research into genetics of resistance and tolerance to livestock disease is rather limited in terms of disease, breeds and species investigated, and parasite infestation will always be there in any farm. So the animals that are genetically resistant to parasite can help to save costs, improve productivity, and reduce drug residue in meat and etc. And new techniques and sources could be used to generate new data for best selection of livestock for disease resistance without compromising production efficiency and product quality. So certainly, genetic selection will not solve all of our livestock disease problems. Therefore, management, nutrition, vaccination, culling and therapeutic treatment and other measures must accompany genetic approach to reduce the impact of livestock disease and parasite on profitability and animal well-being. That's all from us. Thank you.